In this video tutorial, I am going to discuss about nitrogen removal from amino acid. To understand this concept, we have to first look at the structure of amino acid. In the structure of amino acid, there is one central alpha carbon to which one alpha carboxyl group is attached and one alpha amino group is attached. On the third valency, we have hydrogen, and on the fourth valency, we have a side chain R. Now, practically speaking, this alpha amino group, it protects this remaining amino acid from the oxidative breakdown. So, that means if this amino acid, it wants to undergo breakdown or catabolism, then first and obligatory this amino group must be removed. Then only this remaining carbon skeletal, it can undergo further metabolism, right. So, almost all the amino acid, they carry out common strategy to remove this alpha amino group. So, let us first take an overview. Suppose we have any amino acid. What they do to get rid of this amino group? So, first what they do, first they transfer their amino group to alpha ketoglutarate. This alpha ketoglutarate, it gets amino group from this any amino acid and gets converted to the glutamate. Remember, glutamate is the another amino acid. So, amino acid had given their amino group to the glutamate. Then what will happen to remaining carbon skeleton? Remaining carbon skeletal, it will be now called as a alpha keto acid. This alpha keto acid, it can undergo further metabolism, as I told you earlier that once amino group removed, then the remaining carbon skeleton, it can undergo easy metabolism. So, this alpha keto acid can undergo for the energy generation, or it can go for the glucose synthesis, or it can go for the fat synthesis, or it can go for even synthesis of other non-essential amino acids also, right. So, this way it can be further metabolized. This process of transfer of amino group from this any amino acid to alpha ketoglutarate, it is called as a transamination reaction. So, in that way, all the amino group from all other different amino acid, they get concentrated into this glutamate molecule. Then what will happen to glutamate? Glutamate undergoes deamination. So, by the deamination, this amino group from the glutamate, it gets released in the form of ammonia, and the remaining carbon skeleton of glutamate is the alpha ketoglutarate, and this alpha ketoglutarate, it can be reutilized at this step. Now, what happens to this ammonia? This ammonia, most of the ammonia, it gets incorporated into urea and gets excreted. So, excreted is urea, or this ammonia can get incorporated into other biologically useful compounds. So, this box, it represents overview of amino acid metabolism. And here two important processes are that one is transamination and second one is deamination, and in this video we are going to discuss about this both process in full detail. So, let us start with the transamination. So, first is the transamination. What is transamination? As you can see, its name suggests trans means transfer and amination means amino group. So, transamination is nothing but the transfer of amino group. So, transfer occurs between two molecules. So, here transfer of amino group occur from the amino acid to another keto acid. Let us try to understand this transamination with the help of chemical reaction. So, here we have one amino acid. This amino acid, it acts as a donor of amino group, and we have another keto acid, that is alpha keto acid 2. This will act as a acceptor of amino group. So, this amino acid 1, it gives its amino group to this alpha keto acid 2. So, alpha keto acid 2 now gets converted to another amino acid, that is amino acid 2. And this amino acid 1, after losing its amino group, it gets converted to its respective keto acid. Let us say its respective keto acid is alpha keto acid 1. This reaction is called as a transamination reaction. Transamination reactions are reversible reaction and they are catalyzed by the enzyme aminotransferases. transferases. They are also called as they are also known as transaminases. All these aminotransferases or transaminases, they require one coenzyme, and that coenzyme is the pyridoxal phosphate. So, pyridoxal phosphate, which is often abbreviated as a PLP, this acts as a coenzyme for these various aminotransferases. Remember, all the aminotransferases reaction, they are the reversible reaction. These enzymes, they are found in the cytosol or mitochondria or at both the places in almost all the tissue, right. Now, in this transamination reaction, as we can see, there is one pair of amino acid and its respective keto acid and another pair of keto acid and its respective amino acid. In human or mammalian biochemistry, this second pair of keto acid and amino acid, it is almost always fixed. It is almost always this second pair, keto acid is alpha ketoglutarate and the second amino acid is glutamate. So, we can say that this much portion of transamination, it is almost always fixed. So, this is fixed, whereas this portion of transamination reaction, it is variable. And for this variable part of transamination reaction, we have a different aminotransferases. Let us say if this is a glycine, we have a different transaminase enzyme. If this is a valine, we have a different transaminase enzyme, right. So, the specificity of all these aminotransferases, it lies not on this fixed part, rather it lies on this variable part. So, specificity of various transaminases, it is determined by this amino acid which is donating amino group, right. So, specificity lies on this amino group donor. So, for example, if it is alanine, we call them as a alanine transaminase. If this is aspartate, we call them as a aspartate transaminases. So, based on the specificity, their naming is also given. So, as you can imagine, we have a numerous different types of aminotransferases or transaminases, but in human biological system, we have two very important transaminases. One is alanine transaminase and second one is aspartate transaminase. In the alanine transaminase, as I told you earlier, that this variable part amino acid will be alanine. So, we can easily rewrite this transamination reaction for this alanine transaminase. So, here the amino group donor is alanine. This alanine gives its amino group to alpha ketoglutarate, this is fixed. So, alpha ketoglutarate, after gaining amino group from the alanine, it gets converted to glutamate, and alanine, after losing its amino group, it gets converted to its respective keto acid, and here it is pyruvate. Remember, all transamination reactions are the reversible reactions, so they can occur on the both direction. In case of aspartate transaminase, here amino group donor is the aspartate. It donates its amino group to this fixed alpha ketoglutarate. Alpha ketoglutarate, after gaining amino group, gets converted to glutamate, and aspartate, after losing its amino group, gets converted to its respective alpha keto acid, and for aspartate it is oxaloacetate. This is also reversible reaction, and as I told you earlier that we do not have only these two transaminases, we have a numerous transaminases, and all the transaminases along with this alanine transaminases, whenever amino acid catabolism occurs, this is the rule for the transaminases, that whenever amino acid catabolism occurs, this transamination reaction, they work in the direction of glutamate synthesis. So, they work in the direction of glutamate synthesis. But there is one exception to this rule, and that exception is applied to this aspartate transaminase. In case of aspartate transaminase, during amino acid catabolism, this reaction, this reversible transamination reaction is not working in the direction of glutamate synthesis, rather it is working in the reverse direction, that we call it as a direction of aspartate synthesis. So, here there is a direction of aspartate synthesis. So, this thing we have to keep in mind, that all the transaminase, including alanine transaminase, whenever amino acid catabolism is going on, they work in this direction from left to right, or in the direction of glutamate synthesis, whereas the exception is aspartate transaminase. It occurs in the reverse direction towards the aspartate synthesis during amino acid catabolism. Now, it is a time to look at the mechanism of action of these various transaminases. So, now we discuss about mechanism of aminotransferases, how this enzyme work. So, first look at the schematic structure of this enzyme aminotransferases. Suppose this is the aminotransferase enzyme with its active site. Suppose this triangle, it represents its active site. So, in the active site of this aminotransferases, there is one lysine residue, and with this lysine residue there is epsilon amino group, and with this epsilon amino group, this pyridoxal phosphate or PLP is covalently bound. It is bound with this epsilon amino group of lysine residue, and this is the enzyme. Here PLP or pyridoxal phosphate is working as a coenzyme. Now, pyridoxal phosphate is nothing but the active form of vitamin B6. Now, what happens? How this enzyme works? So, let us try to understand how this enzyme works with the help of alanine transaminase. So, we know that in alanine transaminase, alanine comes first and it donates its amino group to the alpha ketoglutarate. But what happens actually? Alanine don't directly transfer its amino group to alpha ketoglutarate. Rather, alanine first transfer its amino group to this enzyme. So, suppose over here we have enzyme, this is active site, this is its lysine residue, and this is the PLP or pyridoxal phosphate. So, alanine first give its amino group to this pyridoxal phosphate, which is covalently bound to this enzyme. So, now enzyme will look like this. This is enzyme main structure, this is active site, this is lysine, and this is PLP. Now, this PLP or pyridoxal phosphate has got a amino group. So, amino group over here, and alanine, now it has lost its amino group. So, alanine gets converted to pyruvate, its respective alpha keto acid. Now, this pyridoxal phosphate along with the amino group, we call it as a pyridoxamine phosphate, and whereas here we have a pyridoxal phosphate. Once this enzyme molecule has this amino group, then this amino group, it can be transferred to alpha ketoglutarate, right? So, this alpha ketoglutarate, it takes up amino group from this enzyme. So, enzyme gets regenerated, whereas alpha ketoglutarate, it gets converted to glutamate. So, and we know that all transamination reactions, they are reversible. So, all of these steps are reversible steps. So, this is the mechanism of action of various aminotransferases. Now, here I want to show you one very interesting thing. That thing is that here what happens, we have a two substrate. One substrate is alanine and second substrate is alpha ketoglutarate, and here we have two product. The first one is the pyruvate and second one is the glutamate. But here what is happening? See, first substrate 1 enters, then this product 1 gets released from this enzyme complex. Once product 1 is released, after that substrate 2 enters, and later on this product 2 gets released. So, it is happening like this. Suppose this bar represents enzyme. First substrate entered, like this, alanine entered. Then after that, this first product is getting released. Once product is released, then after that this substrate 2 is coming. So, it is surprising, right? This product is released before even this second substrate can enter. So, second substrate enters and the second product get released. So, this movement, this down, up, it looks like a ping pong ball. So, this mechanism is called as a ping pong, and as there are 2 substrate and 2 product, this 2 substrate and 2 product, it is called as a bi-bi. Bi means 2. So, this entire mechanism, it is called as a ping pong bi-bi reaction, and this aminotransferases, they provide one very good example of this ping pong bi-bi reaction. So, now let's rapidly revise this aminotransferases. Remember, aminotransferases, they are all reversible, as it is shown by this double headed arrow. There is no any free ammonia liberated, as it is evident from this reaction. These reactions they require pyridoxal phosphate as a coenzyme. As we can see that this amino acid here, this glutamate can be synthesized, or if it is occurring in the reverse direction, then all other non-essential amino acid can also be synthesized. By this transamination reaction, it can divert excess. Suppose we have here excess amino acids, then it can divert all these excess amino acid into this alpha keto acid, and by the alpha keto acid it can go for the energy synthesis or glucose synthesis or fat synthesis, like that. And one ultimate thing is that whatever excess amino acids are there, their all amino groups are transferred to one single amino acid, that is glutamate. So, we can say that glutamate is the collector of all amino acid. And one very important thing, that all amino acid can undergo transamination reaction except for 3 amino acid. These 3 amino acids are K, P and T. These are the one letter codes for the amino acid. K means lysine, P means proline and T means threonine. So, remaining all amino acid except for lysine, proline and threonine, all amino acid can undergo transamination reaction. Now, it is a time to discuss about this deamination. We had completely understood this transamination reaction. So, now we will discuss about deamination. Now, what is the meaning of deamination? As the name suggests, deamination is the removal of amino group. Removal of amino group from amino acid, and this amino group is removed as a ammonia, right. Now, in our body there are majorly 2 types of deamination. One is oxidative deamination and second one is non-oxidative deamination. Majority of the time in our body this oxidative deamination occur, whereas non-oxidative deamination, it plays very minor role, right. In oxidative deamination, we have 3 different types of oxidative deamination. The first and most common and the most major oxidative deamination is the glutamate dehydrogenase. Oxidative deamination by glutamate dehydrogenase, which is often abbreviated as a GDH. Then we have oxidative deamination by D-aminoacid oxidase and by L-aminoacid oxidase. If we look at the non-oxidative deamination, we have 3 different types of mechanism. One is amino acid dehydratase. The second one is amino acid desulfhydrase, and third one is the histidase. Here, out of this oxidative and non-oxidative, this oxidative deamination plays a major role, and with various types of oxidative deamination, the maximum oxidative deamination is carried out by this glutamate dehydrogenase. So, let us first discuss about this glutamate dehydrogenase which carries out oxidative deamination. So, glutamate dehydrogenase which is often abbreviated as a GDH. Now, why glutamate dehydrogenase is the major form by which oxidative deamination occurs? The answer is that earlier in the transamination we had seen that all the amino groups, they are by the process of transamination, they are concentrated into the glutamate. In other words, glutamate is acting as a collector of amino group, right? And this glutamate, luckily we have an enzyme that this glutamate dehydrogenase, it can rapidly remove amino group from the glutamate, right? So, glutamate, by the action of this glutamate dehydrogenase, amino group is released as a ammonia, this is NH3, and remaining carbon skeletal is alpha keto glutarate. But here, as I told you earlier, this is oxidative deamination. So, here oxidation also occurs, and in this NAD plus gets converted to NADH. Now, just like transamination, this deamination is also reversible. So, alpha ketoglutarate can be converted back to the glutamate by the same enzyme, and here in this reverse direction amino group gets added to this alpha ketoglutarate. But surprisingly, in this reverse direction NAD is not used, rather NADP is used. So, NADPH gets converted to NADP plus. So, there is no doubt in that this glutamate dehydrogenase, it is a reversible, it catalyzes reversible reaction. But if it occurs in this forward direction, it is called as a oxidative deamination, where ammonia is getting released and this NAD plus is getting converted to NADH. When it occurs in the reverse direction, everything is same, just one thing is different, that is instead of NAD, it is utilizing NADP, okay, it is NADP dependent. And here in the reverse direction, here on this direction there is oxidation. So, in this reverse direction there is a reductive or reduction. So, reductive, and on this direction there is a deamination. So, in this reverse direction it is a amination. So, this you can so we can say in other words that in oxidative deamination it is NAD plus dependent glutamate dehydrogenase, whereas in case of reductive amination the same enzyme is dependent on NADP, right. So, this thing we have to keep in mind. Now, what will decide that this glutamate dehydrogenase, whether it will work in this direction or in this direction? So, that is being determined by the relative concentrations of the glutamate, alpha ketoglutarate, concentration of ammonia, or ratio of this NAD plus to NADH or NADP plus to NADPH. All these factors will determine that in which direction this reaction will go. Just to give you one example, suppose you have taken just protein rich meal. So, because of protein rich meal, because of the digestion and absorption in our blood, glutamate will increase. So, this increased glutamate, it will lead to direction of this reaction into this from left to right, that is towards the alpha ketoglutarate, right. So, more ammonia get generated. Now, this glutamate dehydrogenase is also under regulation. It is allosterically inhibited by the GTP, that is guanosine triphosphate, whereas this glutamate dehydrogenase, it is allosterically stimulated by the adenosine diphosphate. So, this is how the regulation of glutamate dehydrogenase goes on. So, this glutamate dehydrogenase, it is the major mechanism by which deamination occurs. Now, let us look at the other minor reactions of deamination. Now, let us look at the deaminoacid oxidase. This deaminoacid oxidase, it is the second major deamination process in our body. So, in this what happens? Generally deamino acid, they are not used for the synthesis of mammalian protein, but in our diet it contains so many different D amino acid. So, from the diet our body gets D amino acid. So, this D amino acid must be metabolized. So, what happens to this D amino acid? This D amino acid will be acted upon by this D amino acid oxidase. It is often abbreviated as a DAO. So, it gets metabolized by the D amino acid oxidase and gets converted to its respective alpha keto acids, right. And here with this amino group gets released in the form of ammonia. But along with this, what happen? Oxidation will occur. So, oxidation, this D amino acid oxidase, it depends on the FAD. FAD gets reduced to FADH2. Now, this FADH2, it again convert back to FAD. It takes up the oxygen, and this hydrogen and oxygen, they combine to give rise to H2O2. This is hydrogen peroxide, can do oxidant damage to us. But luckily this D amino acid oxidase, it is present in the peroxisome, and especially peroxisomes of liver and kidney. And in the peroxisome there is one more enzyme, that is catalase. This catalase, it breaks down this hydrogen peroxide into water and oxygen molecule, and just to balance this reaction it is 1/2 oxygen molecule. So, this is how D amino acid oxidase, it causes metabolism of D amino acid. Now, what happens to their alpha keto acids? This alpha keto acid may have a same fate as of our L amino acid alpha keto acid. So, this alpha keto acid, they can undergo for the energy generation, or they can give rise to glucose, they can give rise to fat, or even this alpha keto acid, they can undergo various transamination reaction and can give rise to synthesis of other non-essential L amino acids, right. So, by this way D amino acid can get converted to L amino acid. Let us look at the third deamination mechanism, that is L amino acid oxidase. The third one is L amino acid oxidase. Now, L amino acid oxidase, they play very minor role in amino acid metabolism. Their mechanism of action is almost similar to this D amino acid oxidase. So, L amino acid, they get converted by the L amino acid oxidase to alpha keto acids. This alpha keto acid may have a same fate like this. Now, here amino group is released in the form of ammonia. Here instead of FAD, FMN is used. So, FMN gets converted to FMNH2. This FMNH2 gets converted back to FMN with the help of oxygen. So, this 2 hydrogen and 2 oxygen combines to give rise to hydrogen peroxide, and this hydrogen peroxide is broken down by the catalase to the water molecule and oxygen molecule, right. So, this reaction is almost similar to this D amino acid oxidase with the exception of this coenzyme, right. Remember, L amino acid oxidase, they play very minor role in the amino acid metabolism, and the location is also same, that this also occur in the peroxisomes of liver and kidneys. Now, let us look at this non-oxidative deamination. So, we have 3 simple amino acid dehydratase, amino acid desulfhydrase and histidase. So, let us look at first amino acid dehydratase. First, amino acid dehydratase. This amino acid dehydratase, it works or it has action on hydroxyl group containing amino acid. So, we have a serine and threonine as a hydroxyl group containing amino acid. So, this enzyme, it works on this serine and threonine, and it causes the removal of ammonia, amino group as ammonia, and they get converted to their respective alpha keto acids. The second type of non-oxidative deamination is the amino acid sulfhydrase. sulfhydrase. This works on cysteine and homocysteine. By the action of amino acid sulfhydrase, the cysteine or homocysteine, they undergo non-oxidative deamination and gets converted to their respective keto acid, and in this case it is pyruvate. Now, you might be wondering that in case of alanine also the respective keto acid was pyruvate, and in case of cysteine and homocysteine the respective keto acid is pyruvate. But here what happens, not only ammonia, this amino group as a ammonia is removed, but both are sulfur containing. So, their sulfur is also getting removed as a H2S. So, we get this keto acid as a pyruvate. And the third and last mechanism for the deamination is by the histidase. As the name suggests, histidase acts on the histidine. So, histidase act on the histidine, and here amino group of the histidine will be removed as ammonia, and its respective alpha keto acid is urocanate. So, this is how oxidative and non-oxidative deamination occurs. And as a summary, see what happens. First, transamination occurs. So, suppose this is the transamination. In the transamination, we had seen that all amino acids, they donate their amino group to alpha ketoglutarate, and alpha ketoglutarate gets converted to glutamate, and this amino acid, they gets converted to their respective keto acids. After transamination, there is a deamination, right. In the deamination, the major deamination mechanism is oxidative deamination. In that, what happens? Glutamate gets converted to alpha ketoglutarate, and meanwhile amino group is released as a ammonia. And during this reaction, NAD plus gets reduced to NADH. So, as you can see, this transamination and this deamination both are happening in the coupled manner, or both are going in hand, and by that this amino group, this nitrogen of amino acid is successfully removed. So, transamination and deamination both are coupled process, both are dependent on each other. So, because of that, many author refers both of these processes in combination, and they label them as a Transdeamination, because these authors, they want to emphasize that these both processes are not a different process, but both are just a two portion of one larger process. Okay, but if you look at them like a transdeamination, or if you look at differently transamination and deamination, once concept is clear, nothing can bother us. And I hope that everything about transamination and deamination is clear. Thank you.